15. So now the first thing that we have to remember when we talk about the effects of variance is this graph that I briefly showed a few times this year. Let me just repeat it. This is number of residues aligned, percentage sequence identity, pick a point, 100 residues aligned, more than 30 identical, we are in the blue region. These two proteins have a similar structure. That means in turn you can change 67 residues and not change structure, right? There's a lot of, let's call it, resistance against change. So when you look at this plot, you may have the idea that most changes will be somehow neutral. And this is somehow misleading because what I show here is this is the plot that we get for proteins that evolved in nature. What's so special about proteins that evolved in nature? So why could there be a difference between the way proteins evolved in nature and they could in the tube, yes?
Because proteins that evolved and got useless afterwards?
That's the important point. Everything where the change is not beneficial or neutral is sort of thrown away. Okay? Anything that doesn't work, you will not see in this plot because it will not survive in evolution. So what we see is a very special selection record. And that in fact is why this information is so important. In this selection, that's a selection by function by structure, right? That's why there's some powerful information there. Let's get back to the genome issue. So what types of sequence variations are there? So the simplest one is the substitution. So you flip an A to a C. The next issue is, so we have an insertion, we have several that we insert, or the other way, the flip side, we have deletion, we cut out some letter. There are duplications, so we essentially repeat the same letter here twice. Should have put that in red, I'm sorry. And then there are inversions. So inversions are simply, you read the string the other way around. And you look at the complement because there is a pairing of the 4 nucleotide sequences. Essentially, this is the complement of the other one. So these are the typical kind of changes you see on the level of the nucleotide sequence. On the level of the protein, we first of all have two different ones. We have non-synonymous and synonymous. What it means essentially, the nucleotide change. You have changed a single nucleotide, and that may change the amino acid or not. It will not change it. So remember, there are 3 nucleotides that code for 1 amino acids. And so there are different solution things. 4 by the power of 3 is 64. You have 64 possibilities, but you have only 20 amino acids. So some amino acids have more than 1 triplet. Codon, so-called codon. And that means that it could be a change that is sort of neutral. And in fact, that does happen. And they are called synonymous, silent. You don't see them on the level of a protein sequence. And then there are changes that really matter. Now, what in this lecture we will be concerned with essentially are only the ones that change the amino acid sequence. A change in the nucleotide sequence that changes the amino acid. Now, there's what is referred to as nonsense changes. So these are changes that lead to a premature stop. What that means is that when you read off the genomic information, the read is stopped and not the entire protein is expressed, but only part of it. Okay? So then you get a chopped protein, so to speak. Then there are insertions, deletions, and duplications, very similar to the idea on the DNA level. And there is something, the inversion can lead to sort of corresponding amino acids. But that is sort of means something else in the protein world. It's just another amino acid that comes from the inversion. Now, to get to words, typically today people talk about SNFs as single nucleotide variants. So this is you change 1 letter. And on the level of the amino acid sequence, if you change 1, people talk about that as SAF. Then there is sort of mutation. That is essentially a change that may be disease causing, essentially a SAF is a mutation. I'm going to refer to 19 non-native. What that means essentially, if you change at one particular position the native amino acid, so that's the one that you have there, against all the other 19, against all the other 19 that are not there, right? That's the non-native one. And then, well, let's keep that. When we look at what mutations do, I said that the evolution in that we see that there's a lot of resistance against change. Here's one example, and in some sense this is the mother of all mutations, or SAFs, because one was the one that was first discovered and described. Hemoglobin is a protein that carries iron. It's important for breathing and for all kinds of things. And in hemoglobin aggregates upon deoxygenation. And here in this particular protein, if you have one amino acid change, then it leads to the case where you aggregate. These aggregate cells are shown here. This is sort of the normal one. These are the aggregate cells. So one single amino acid change, the thing aggregates and it follows a disease, right? You have that variant, you have a disease. Now, how could you possibly try to predict the effect of a variant? Now, the effect, here's the next issue. What do we mean by effect? Do we mean that it changes the structure? Do we mean that it changes the function? Do we mean that it actually changes the organism? So I talked about a disease. For the disease effect, it's important that this change does something that I will feel on my body. Many changes on the level of the way the protein functions may be that the protein functions just less well. And I will not sense that as a disease. I will sense it as I'm less fit to carry out some certain task maybe, right? So these are different things. We will focus on this on the molecular level. So for us, change mostly, the idea is upon molecular function, the way the protein binds. So the most dramatic way in which you can change function is you make a change in the binding site. So for instance, you need in the binding site a positively charged residue, you flip that positively charged residue to a non-charged residue, that will affect the binding, simply because the binding depends on its charge, right? Again, the problem is that there is a high resistance. The first method happens to be called SIFT. And I just put up the acronym because it doesn't give you any idea how the authors could have done it. The more direct way really is using the Blossom matrix. Is the idea, and I should have written the Blossom matrix more clearly here. So the Blossom matrix simply tells you what in a set of related proteins where I do not know the effect of the variant, I just know that they are related, what can I change against what? That is essentially what the Blossom matrix tells you, right? And then we could simply look at this Blossom matrix and say, well, if Blossom says this is sort of a neutral change, and suddenly you're completely right, because in principle the Blossom matrix reflects the biophysical features. It's just I don't have to sit down and say hydrophobicity is charged, blah, blah, blah. And again, I do not know which one, how far the other one would have gone, or labeling some binding sites. Maybe this would have been a very interesting alternative that is also as simple in principle. Anyway, so this was done by the group of Steve Hanikoff, Paul and Nick, and so Steve Hanikoff is the one from who this Blossom matrix comes, so it's no surprise that for them this was a much simpler way of thinking. And that in fact is the first method, and that is still used today in principle. Works fairly well. But there is something that is missing in that step. And that something is sort of brought forward by this method here. So what I mean here is ultimately, and again I should have blown this up here, it's the alignment. When you align one protein with a family, then you see in that protein which residue has to be conserved, which residue cannot be or does not have to be conserved, right? So for some residues, no matter how long the protein is, there are some residues that are entirely conserved. Changing those will have more of an effect. Yes, the Blossom matrix has that as a feature on average, but it does not have that for any specific protein family, right? Remember, you were in last semester, so the profile-based alignment method. This Blossom is the pairwise alignment. Then you build up many pairwise alignments and you build a profile. And this is exactly what is missing here, the profile information. Yes?
But like, with all due respect, this is kind of a simple machine learning.
Oh, that is not a machine learning at this point.
Well, like in a very generalized terms, calculating alignments is kind of machine learning.
Because but you mean the alignment itself is machine learning?
Yeah. The process it's very similar to k-nearest neighbors.
Yeah. Okay, fair enough. Point taken. Yes.
Yeah. So
it's a little bit of a push, but point taken. Yes.
So when you said no machine learning, like
I don't optimize any parameter.
Okay. Yeah.
Well, in principle, I could do that.
Yeah.
In the simplest iteration, and again, I'm going to show you on Thursday a quick result. What would happen if you just use one parameter. So yes, you do the alignment, and that sort of has an optimization process in itself, but optimization on some other task, okay? So you solve the alignment problem, and then you run the alignments, and then you just say, conserved, yes, no. One parameter. And that is very, very atypical to machine learning, right? You optimize one parameter. And you can do that on a tiny data set. And that ultimately is sort of the major part of polyfen here, a very clever way, in fact, not only using the simplest way, but a clever way of doing it. SnapS3D uses on top the information for proteins for which we know the structure. They say, well, whether it's conserved inside of a protein or conserved outside has a different meaning. And they take that one into account. And those are sort of the simplest first three methods that were available. And again, they all do not really optimize parameters, so these methods are available at a time when you do not have enough data. So you have some incidental data about some proteins or some variants that have effects, such as the one in hemoglobin I showed you, but that's essentially all you need. And that is all you need because, in fact, it is complicated to look at a large data set because effect upon things differ really on the level of, in this particular case, you see a big phenotype difference. You see the two mice have different colors, right? Something that you immediately see. So you see an effect of a variant on an organism. In many other cases, you will possibly see a differential binding, right? That's a much more fine-grained effect. In other cases, you will see that the structures change. Now, are all of these effects the same? Somehow they all affect function, or somehow this is very clear that these mice change, or that on the large organism change, or the organism changes here because, in fact, there's a molecular change. But not all molecular change the organism, right? So can we integrate all of them, and how do we integrate that, and how do we do that? It's not that trivial. Initially, so there was a database that collected it. That database was sort of stopped a few years ago. And in this database, we have essentially 40,000 effect variants and 14,000 neutral variants. So the first statement is, this is for all the organisms that we have, and that is SAVs, so it's 40,000 SAVs, 14,000 SAVs, and that is integrated over any kind of effect we have. So some of these effects are really measured, and some of these effects, there's somebody saying, well, this looks different from that. Okay? And they're all part of the same database. There's no qualifier. It's just a binary value. Okay? And you immediately see that this database has issues. And despite all of these issues, we have a relatively small data set. Why do I call that relatively small? Well, just look at what you have in human. So you have 20,000 proteins. Each of these is sort of 400 residues long. Let's make it 500. So 20,000 times 500 gives you an idea about the number of residues we have, which in fact is 10 million. Is that true? 20,000 times 500? 6, yeah. 20 million, 100 million, 10 million. And then for every single one you have 20 possible variants. So it's a huge space. Right? 200 million. And behind that, this is a small number. And again, if you now did that for everything, the next issue is the imbalance. It's sort of a quarter that has no effect. Now, we do not believe that this is the sort of true value. Well, in some sense, we assume that it depends really on your scale. Every variant is going to have some effect. Sometimes the scale may be so minute that you will never be able to measure it. But how many of the variants have a strong effect? Is it true that about only 1/4 of the variants have a strong effect? Most likely this is not true. So why is that what we see? Okay, so there's a high experimental preference for effect simply because it's easier to detect effect than to detect neutral. Now when we train devices, most likely somehow we will get a bias toward effect. And somehow we want to sort of get out this bias. So we want to somehow train something where neutral at least is a major class. So I have to create some data set from in silico. And the idea was to simply look at proteins for which I know the enzymatic function. So let's look at this example. So I have two proteins here, one and the other one. And I know experimentally both of those enzymes. I know exactly they have for both we have experiments, independent experiments that say they do exactly the same thing, these two proteins, right? And you immediately see for wherever they differ, that must be neutral. At least, okay, it could be another function that is affected by these differences. But if I assumed that most of these two enzymes do is carried by the enzymatic function that I can measure, and it is the same between the two, then I would argue that where they differ is neutral. Okay? And this creates a data set that is in the ballpark of something like roughly equal. Why is this not making the neutral number shoot up much more? Because we have to be very careful here. What we look at, we need experimental evidence for both. They should not be too different, and they should not be too similar. The two, and that gets us, and then we need a redundancy reduced version, and then we get to a relatively small number. And that gets us to what I'd call the second generation of methods that use larger data sets and now can do what I would really call machine learning devices. It's still nowhere near deep learning. The deep learning sort of starts in the third generation data sets, where it's also about doing new things with it. And one of the first methods, or the first method, really came from Jana Bromberg. SNAP, currently we have SNAP version 2. And SNAP essentially uses a neural network. I'm not presenting much information about it on the next slides. Essentially, it's a neural network that puts a sequence of 13 consecutive amino acids, just like we did for the prediction of binding sites, and puts additional information. So it looks at the profile, the amino acid variation within a family. It looks at predicted secondary structure, solvent accessibility, and other biophysical features. Some of those predicted features, binding sites that we talked about are put in there. DNA binding sites, a variety of binding sites. And then comes something that is very relevant here, a feature that we have not used before. So since essentially we are putting in, say, 3 consecutive residues, and we want to predict in this one whether mutation of alanine to cysteine, for instance, has an effect. So we're going to put in a 4th residue somehow. We are going to put in something that reflects the change. And the same is true for predicted secondary structure, for predicted biophysical features. For whatever we have, we are asking how is that feature changing upon that sequence of change, right? So delta features. Is it clear what I'm talking about? No. So far, for instance, when we predicted whether a protein has a PP binding site or not, okay, then we used a consecutive window. In this particular case, let's say the k-mer is 3. Then we used for every single position here 21 input units, okay? This was always a 21 input. And then I sort of threw in secondary structure. So secondary structure again was added 1, 2, 3, because secondary structure comes in 3 states, added 3 units to every one of these input units. So instead of 3 times 21, adding secondary structure would add 3 times 24, right? So far, so clear. So that is clear. And then I essentially predict for every single residue the secondary structure. That's the way it is. Now, I have a variant, a sequence variant. So I have to put the sequence variant in. So I have to essentially put the sequence of what it was and the sequence what it went to. So that means I'm not having 3 times 21, but I have 4 times 21. Okay? Now when I look at secondary structure, I would then have 4 times 24. You're still with me on that? Okay, let's go back. So this is PP prediction. I'm sorry for the whiteboard. And now we do SAF effect. And again, we look at 3. So sequence only is 3 times 21, so 20 amino acids plus one spacer, plus secondary structure here would give us 3 times 24, because secondary structure comes in 3 states, sorry. Now for SAF, instead of on the sequence only side 3 times 21, we need 4 times 21, because we have the residue that it's changed into. And I have to represent, I could present that differently, but in the simplest one-hot encoding way, this would be 4 times 21, right? So far so good, or not? Is anybody bumping out at this point? Good. Now I add plus secondary structure here, and this would be 4 times 24. Still the same. Still nothing new. Plus, now comes the delta feature. And the delta feature essentially does not say, okay, I have the version with the A and I have the version with the C, predict secondary structure for both. It says on top, what I'm really caring for is how does secondary structure change. So predicted secondary structure of one, of the native, minus predicted secondary structure for the other. Again, the machine could learn to do this differential, but it turns out to be very helpful to put the information in simply because we don't have enough samples. If I had a very huge data set, most likely the system could sort it out. But for limited sets as we have them, and very, very limited sets, it turns out that these delta features are absolutely crucial. Clear? Okay, so I'm going to just run through a few other methods here. Here's one, mutation assessor, that comes from Boris Reva and Chrysander's group. And the point, the main idea behind that thing. So we have an example where we have an E here is conserved, and then it turns into an N. So the way you see that is in this column, you could have E or N. Okay? This is what I introduced so far through Polyfen. This is what I will do in the machine learning device. But they said, listen, this is not quite true. For the next one, it cannot be either E or N. Whether it can be E or N depends on whether that is sort of the subsequence that belongs to this group here, or the subsequence that belongs to that group. Because actually what you see is that the first 4, the next 3, they differ. In fact, there are differences. The swap from E to N here comes along with some slight, minute functional change. That is why it's a subgroup. Now, that means that I cannot just add up what's the probability in a particular column to, is it E or N? It is very different whether we have a case such as the one that I show there, E, E, E. Yeah, now I write it like this, or whether I have one that is E, N, E, N, E, N, E. Those are very different situations. In terms of what we have discussed so far, they are identical. They're not distinguished. And that is sort of the major step introduced in mutation taster. And again, putting that into a machine learning device is what is currently being used on this one. Here is another idea, and that's the idea of evolutionary couplings, briefly introduced in the context of contacts, originally published by Thomas Hopf in the group of Debbie Marks and Chrysander. So the idea is the following. So we have 2 columns here, so 2 amino acids, and particular points in the alignment, and they sit close in space. Since they sit close in space, what you will observe at these 2 different positions here depends, so what you will observe here depends on what is there. Simply because they evolve as 3-dimensional objects, and these 3-dimensional objects are coupled. They're coupled through the contact. Okay, they're next to each other. Next to each other means that anything that sort of will disrupt this one will affect the binding. Or if in a most extreme one, so if we are in a dense space, if this swaps from a small amino acids to a larger amino acids, and this does the opposite, from a large to a small, you still occupy the same volume, and you could still make this contact without any disruption. If the other one stayed the same, you would disrupt it. That means that you possibly would see switches like essentially like this one here, where you would see that maybe there's some region here where they all have a positive and here a negative, and then they switch to, well, in this particular case to polar, and there they switch too, right? So they compensate that change. And the entire idea behind evolutionary couplings is to sort of inverse this problem and find those and use correlations on this level to infer couplings and use these couplings then as input to predict essentially the effect on amino acid of SAFs for sequence variants. You simply say if this varies and it's coupled to nothing, then it's fine. If it is varied and there's sort of a compensating variant, then it's also fine. But if both of them are not compensating each other, then it's not fine. And that is in fact one of the best methods out there. The problem with that method is it takes a lot of time. It is not easily run for all proteins because it needs huge alignments. How do we compare methods? And this ultimately, essentially, I'm bringing in I'm bringing Snap now. And now, I'm sorry, I forgot that I have this slide in here. So this is exactly what I said. You have the 13 times 21 amino acids, and then there are some other input features that I have. Sorry for the animation. Biophysical features, alignment profiles, probability of residue triplets. That, in fact, is the statement that you had already. Solvent accessibility, secondary structure, residue flexibility, and then some from annotations. You use all of this and you predict SAF effects. Essentially, the score is not binary, but it's sort of projected onto a binary score by having a slider. Now, the overall performance, one way of doing that is there was when we did this, there was a bunch of 1, 2, 3, 4 proteins for which more than one variant was known. So we had for each protein a bunch of measurements, and we compared sort of essentially three different methods, and we simply here have the overall accuracy. The overall accuracy says how many of the variants are correctly predicted to have effect, not effect, binary. Simply, that was in the toggle idea, right? Before this idea here is only a year or two old. And so the problem with that one here was this is only 4 proteins. So how would it work for something else? Then we had the larger data set. On the larger data set, we had some ROC curves where we essentially have here precision versus recall, and we have precision for recall versus recall for the effect and for neutral. So this is typically not called. This is what is this called? Whatever. So there's a different word for that for the negatives, right? You see now 3 different data sets: red buried, blue exposed, and then intermediate. And you see that the behavior, so the red, where red and blue is swaps between neutral on that right side and between effect. So for the effect prediction, we do best. The one that is highest here is for buried. For the neutral, it's just the other way around. We do best for the exposed. There again, so we dial through this curve here by looking at stronger predictions. These stronger predictions allow us to sort of have a reliability index, and we see that stronger predictions are predicted much better. And that, in fact, is also when we classify effect of a variant, we see that the most severe ones are predicted with the strongest score. Okay, so far so good. What we do see is, for instance, annotations help. So with annotations we get overall a better Q2, but in fact we have so few annotations that it doesn't really matter. Now, in some sense, what I've focused on so far is always this Q2 measure. And there are a bunch of major problems with the methods we have. So one problem comes from using something as simple as Q2, because we actually don't know what the ratio between neutral and non-neutral is in the nature. And the Q2, fraction correctly predicted in a binary state, effect or not, is in fact highly dependent on my data set. How many neutrals do I have in my data set? That's very crucial. So we need other measures. The moment I put in other measures, I believe I got the slides out, we get these very long tables, and it's very difficult to have a clear overview. That's the first problem. The next problem is, we at this point, I said, have about, say, 40,000 plus 16,000. We have about 60,000 amino acid SAFs for which we have some experimental data. At this point, I believe we have 200 different methods, and they all use these 60,000. And they're all optimized on the same data sets. I also told you there are 100 million in human alone. So what we have is a tiny subset of what we want to have. And we see that these methods agree very much on the data that we experimentally have. And they actually do not agree very much on all the residues for which we don't have data. So where we don't have experimental data, we cannot say that one is better than the other, but we can measure whether they agree, right? And in that, we see they differ a lot. And ultimately, this leads us to this issue that the methods have all been very over-optimized, and the field has come to a standstill in some sense, or into an impasse, because we all sort of optimized a little bit epsilon, a little bit better of the same sort of thing. And we need to find a way out of it. Somehow there's the hope that we get out of it at the moment because there's just more data. But it's clearly not, the more data is still nowhere near the level of what we want, the 100 million kind of thing. More data means we get a few hundred thousands, but that is all we have. Anyway, so the point that I just made is that there is a lot of identity in the data set between methods, and that methods sort of over-optimize on the same data set, and that means we should possibly find a way to sort of look at where they differ. Yes, the Q2 here is somehow different between these four methods, and yes, the three that were developed are clearly better than random, and the machine learning tends to be a little bit better than the ones that did not use machine learning. So again, this optimized thousands of parameters, and these essentially optimized one parameter. So they are very, at least in this application here, there's a polyfen 2 that used machine learning. This one didn't. It's absolutely a simple method, right? And now, the next question is, what if we looked at all the SAFs that they all predict the same? When I say all predict the same, I really mean only at least two methods predict the same. Let's just kick them out. Let's say if two methods predicted the same way, it's simple. Whether it's predicted as neutral or as effect, I don't care. If methods agree, I throw it out. Let's just look at the set of predictions where at least two methods disagree. So not all methods say the same. So let's only look at the ones where at least two have a different answer, okay? And that is that set. So for that set now, again, two methods disagree. That's the set of SAFs with effect or neutral, for which at least two of these three methods disagree. You see that polyfen and SIFT perform below random. Now you could sort of provocatively ask yourself, how can that be? How can a method perform less well than random? Well, the answer simply is because I removed everything where they work, right? It's totally unfair. So I'm completely twisting the story. The good news is that, in some sense the bad news, if I flip between the two, you see that SNAP performs much less well. So it moves from something like around 80 to something around 60. But it now, for that set, where things disagree, if you look at disagreement and you need a tiebreaker, then that is the tiebreaker. And that is sort of a major difference, right? This is, for things where they disagree, this is the only method out there that is better than random. And that sort of is another way of looking at it under the same score. That's very, very important for users. In the way you use that method, you simply see disagreement, you apply that method, right? That's as simple as that, and you have a big gain from doing that. Okay? So then there's sort of next generation, Snap 2, came from Max Hecht. he sort of introduced new positive, but also new negative, ended up with a sort of 100,000 data set, and introduced more features such as contact predicted by coevolution, essentially the feature that I talked about in a much simpler way, and overall this thing does slightly differently, it does slightly better. And I believe I'm going to have a short break, a short talk today. On Thursday we're going to talk about what we can now do with these predictions, some examples, and in fact what we in some sense can learn for various questions. And then Dimitri will talk about how we can try to apply the deep learning to sort of go for the next step. Thank you for your attention.